Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking a look at animal development. This is going to be kind of a continuation of our last one on reproduction. This is our second to last video in the organismal physiology series, so hang tight, we're almost done. Let's talk about the stages of development. There's kind of two main stages. You've got embryonic development, and that's all occurring before birth actually happens. Start off with the zygote after fertilization. Then you have some cleavage, which is splitting of the cells. You turn into a blastula, eventually a gastrula, and then you become an embryo. And then birth occurs. Now, this happens differently in different species, so there's no set rule on that one. But then development continues after birth until finally you reach the adult full maturity stage, and we see that with pretty much all animals. Almost no one is born, fully developed. So the amniotic egg, let's talk about that for a moment. That is a critical evolutionary feature, right? It allows a full life cycle to occur on land, and that is what we find in reptiles. And then the amniotic egg gets passed on to birds and mammals from there. The amnion is the part that is actually protecting the actual embryo itself, the fetus. So that's the blue section right here. You've got the chorion, which is going to help facilitate gas exchange, right? Because you want to be able to bring in oxygen and get rid of CO2. That's something we don't necessarily think about with eggs, is being able to facilitate gas exchange, but they do. Then you've got the allantois, which is this section over here, and that's where waste is stored because you're going to have nitrogenous waste products being produced during the growth process. You need somewhere to actually put it. And then you've got the yolk, which is right here, and this is a food source for the fetus, so that way it can grow while it is inside the shell. So, starting off, we have the process of fertilization. In sea urchins, that is an external process. So when a sperm encounters an egg cell, it is going to release an enzyme that breaks down the jelly coat. And the reason why they have this jelly coat is to protect them, right? Because the eggs are, floating around in the water. So you need to have some sort of protection while you're there. So those enzymes are released from the acrosome, and they break down, and then it allows an actin filament to be able to bind with some receptors that are on the outside of the egg cell. When the receptors bind properly, so this is one of those types of the mechanical isolation, right? We talked about the fact, or sorry, gametic isolation. We talked about the fact that only certain sperm could meet with certain eggs. That is this process occurring right here. So if the match happens, then there will be fusion between the two. When the fusion occurs, that is going to release these cortical granules, which push out the fertilization envelope and basically make it so that it is impossible for another sperm to be able to penetrate and fuse with the egg, thereby ensuring that you only have one extra haploid cell that is added, extra haploid set of DNA that is added, so you end up with your 2N cell. Now, this occurs almost instantaneously, and so we call it the fast block mechanism. And that makes sense, right? Because you're in an external environment, there's probably a lot of sperm around. Now, in mammals, this is an internal environment where fertilization is occurring, so there's no jelly coating around the outside. You have some follicle cells that are kind of hanging out that the sperm has to work its way through. Again, fusion is going to cause an impenetrable space to form, although the way it occurs is a little bit differently. So fusion of gametes in a mammal triggers a release of a calcium wave, a calcium ion wave, and that calcium ion wave is what forms the fertilization envelope, and it prevents other sperm from entering, just like it does in the sea urchins. Now, this takes about 30 seconds rather than it happening almost instantaneously, so we call this the slow block. It takes more time. Can we just pause for a moment and talk about how much calcium is used in? I mean, we've seen it used in all kinds of different things and all kinds of different processes. So, really kind of shows you how important calcium is, and to making sure that you're getting enough calcium in your diet. Just all the different things we've talked about. So here we go. Here's our zygote. This is a single-celled urchin. You can see here is the cell itself, and then around the outside, this is that fertilization envelope that is going to help prevent fertilization by a second sperm cell. Then we have cleavage, which is the splitting. So here it has gone through two different cell divisions, and then it's continuing to go through cell divisions here. You can see that the cells themselves are getting smaller as time goes on. So there's not actually any growth that's occurring at this time in terms of it getting bigger. It's just splitting, and as so the cells are getting smaller. So this is a picture of some frog of a frog zygote, and you can see the different types of cleavage that are going on the different cells. I really like this picture because these pictures just because they just show you, real up close and personal kind of what it looks like when the cells are all jammed together. At this point, each of these cells are totipotent. If you recall back to when we talked about stem cells, that means that each of them can become a brand new organism because they can each turn into any other type of organism. And if at this stage we have some sort of splitting, so let's say some cells get broken off during this point, you are going to end up with a genetically identical second embryo that will grow, and that is where you get identical twins from. So pretty cool. So here we go. The more Rula is the next stage. You can see that we kind of have this space in the middle with the cells around the outside. The blastula has a mass of cells that kind of conglomerates on one end of the cavity that's in the middle, and then we have gastrulation. Gastrulation is going to produce a gastrula where we are going to begin to form the primary germ layers. If you will recall, the primary germ layers are the endoderm, the mesoderm, and the oh man, totally forgot the ectoderm. Sorry, the ectoderm, the mesoderm, and the endoderm. Wow, totally had a mind blank right there. And all of those organs and tissues are going to develop from these three layers, and we're also going to form the primitive gut tube, which is also known as the archenteron. So this is the process of gastrulation that is in the process of occurring. You can see that you kind of have this mass of cells along the outside, and then it's kind of bending in on itself, which is kind of interesting. So what happens is that you have these poles that form on either side of the cell, and the poles that form on the different sides of the cell end up giving you kind of the two areas. One of the areas is going to begin to form the blastopore, which is where the cells begin to kind of fold into each other into the center. And you can see that right here, the blastopore forming. And then the other end of the cell is going to become the other side. So one end can become the anus, and the other becomes the mouth. That is dependent upon whether or not you are a protostome or a deuterostome. And it's not, terribly important that you know the differences between those two, but just, it can go either way depending on which side you are in. Frog gastrulation occurs in a slightly different way. So rather than it folding in, the gastrula actually, or the blastocele and the archenteron form from the inside out, which is a little different and kind of interesting. Don't worry about all the details on this. We're just kind of showing you some different ways that this can occur. So here's kind of that early gastrulation process occurring where it's folding in. Again, this is a sea urchin. Here's the gastrulation process continuing to occur. And here we've got it's been totally gastrulized. You can see that it's kind of all the way through, and we're beginning to form some different structures of the animal. And then here you go. Here's the full larva. It is now, in that stage where it would be considered to be born. In this case, although has been developed in the water, so it's not quite the same. All right, different germ layers. So we've kind of talked about this a little bit and kind of shown you where they come from, but they all turn into different parts of your body. So the ectoderm turns into your ectodermis, your epidermis, your skin, which makes sense. Ecto means outside. It turns into the epithelial lining of the mouth and the anus. Turns into the cornea, lens of the eye, your nervous system, the sensory receptors, adrenal, medulla, tooth enamel, and the epithelial of the pineal and pituitary glands. Your mesoderm turns into the notochord, your skeletal system, the muscular system, the muscular layer of stomach and intestines, the excretory system, circulatory and lymphatic systems, reproductive system, dermis of the skin, lining of the body cavity, and the adrenal cortex. And then your endoderm turns into everything else. So that's your digestive tract, the respiratory system, linings of the reproductive systems, your liver, pancreas, thymus, thyroid, and parathyroid glands. So, there's a lot of things that come out of all these different layers. It's not terribly important that you know all of them. I'm just trying to give you all an idea of kind of what's going on and where these different things come It gets a little complicated. The couple things you should know, which is neurulation, which is development of the primitive notochord. And remember, the notochord is the precursor to the spinal cord. And then you've got organogenesis, which is the development of organs, and those come from somites, which are patches of cells that are eventually going to develop into specific organs. How did they figure that out? Well, what they do is they dye specific cells, and then they follow those cells throughout development to kind of see what they form, and then over time, slowly, these different patches of cells differentiate and form the various organs and all the different types of tissues that we find inside our body. Morphogenesis is the changing in the shape. So morpho, shape, genesis, creation, right? So the creation of different shapes. And that occurs just throughout the development process. It is critical to development, right? Because think about form and function. We've talked about that several times. You've got to have the right form in order to be able to perform your function. So there are a lot of different ways that this can occur. We can have microtubules and actin filaments that can change shape, move, interact with one another in order to create different shapes. We can also have cells. The cells can be next to one another. They can then kind of converge. And when they do that, they extend out, they produce growth, because now you, instead of having just 3 cells, if you have them come together, now you have 6 cells. So pretty interesting stuff. All right, let's get to the really good stuff though. Let's talk about human development. We talked about this a little bit last time when we talked about reproduction. So this whole part is familiar. It's what we've been talking about. You've got ovulation and then fertilization, and then cleavage begins. Cleavage is going to continue as it travels down the fallopian tubes. And then we have implantation, where the blastocyst is going to enter into the uterus. Implantation is extremely important because without it, the embryo would not be able to survive very long. It's going to run out of nutrients. And also the endometrium is going to be shed because the menstrual cycle is going to go carry out its course, and eventually when it does so, if the embryo has not implanted, it's going to flush out the embryo with it. You can see a lot of the various parts of the amniotic egg in the embryo's implantation. You can see the amniotic cavity. You can see the yolk sac. All those things are going to continue to develop, and they're going to develop into the placenta. The placenta is a temporary organ. It is extremely important because it helps to facilitate fluid exchange and therefore nutrient exchange between the mother and the fetus. Very similarly to how the amniotic egg has places where it takes away waste and then gives nutrients, that's exactly the job of the placenta. The job of the placenta is to provide nutrients, oxygen, sugars, all that kind of good stuff for the fetus, while also taking away its waste products from the growth process. Now, there is a blood barrier here, so there is never actually any exchange directly between the fetus and the mother in terms of the blood swapping over, which is good because they could be the wrong blood type. So you don't want the immune system of either one attacking the other. So what happens is that you have these capillaries that are formed that are from the fetus, and they are there to help, increase surface area, as we talked about before. And the mother's blood is actually just kind of let out into this space where these capillaries are located, and then it is drawn back in. So it facilitates the exchange across that barrier. Again, the blood cells themselves actually remain separate. Fetal development. It's kind of interesting. We tend to think about growth as kind of being a steady thing, but that's not really true. Most of the growth actually occurs in the last trimester. At 20 weeks, the fetus is only about a soda can big. So that's 20 weeks is a pretty long time. But what you can see here at 20 weeks is that there's a lot of development that has occurred, right? You've pretty much got what looks to be a human in terms of it's got all the features that we would associate with a human. Eyes, a head, fingers, like all that stuff has developed. So a lot of the energy has been put into actually differentiating the cells, producing the various parts, which will then be grown towards the end, or will grow towards the All right, so let's talk about the labor process. Labor is started off by estradiol and oxytocin. Estradiol, which remember is a form of estrogen or is a type of estrogen, is produced in the ovaries and it induces oxytocin receptors on the uterus. So it basically creates a place for the oxytocin to be able to bind. Now, once the oxytocin is able to bind, it is going to both stimulate uterus contractions as well as stimulate the placenta to make prostaglandins. The prostaglandins are going to stimulate more contractions in the uterus, which is going to stimulate the placenta to make more prostaglandins, which is going to stimulate more contractions of the uterus. More contractions of the uterus also form more oxytocin. So you can see how this is going to be a huge positive feedback loop where it is going to build and build and build until eventually we are going to have birth. When birth occurs, the cervix, which is the kind of the gateway doorway between the uterus and the vagina, is going to dilate, and that is to allow the baby to pass through. Once it has dilated fully, contractions in the uterus are going to help expel the infant from the uterus, and then the placenta will follow because it is, remember, a temporary organ, and so it is no longer necessary. So the placenta, along with the umbilical cord, are going to be um, expelled directly after the infant. All right, now we can mess with the development process, okay? And it's actually surprisingly easy. And the way you can do that is you can take pieces from the embryo or from the zygote or the blastula or different places, and you can take them and add them onto different areas, and they will develop as you would expect them to do if they were in their original area. So, for example, they can take pieces of the blastopore dorsal lip, and that is what turns into the notochord. If you take that and stick it onto a gastrula, it will develop into a new notochord. So you can actually get a notochord on either side of um, that embryo, and you can basically, mess with the development of either one, um, which is kind of a weird thing to think about. You can actually create kind of like two embryos next to one another, two fetuses, and it's strange. Anyways, we can also take the blastula and we can rotate it and we can tie things off. So if you were to cut it in half at a particular stage of development, you can end up with two normal embryos or developments. However, if you rotate it 90 degrees and then cut it off, you end up with one that doesn't develop normally and the other one does, right? Because it doesn't have all the cells that it should end up with. That gray crescent area has a very important function in terms of helping to denote the head and tail of the animal. So again, very easy to be able to mess with the development process. And then finally, you can take areas from a limb bud. So that's where the limb is just about to begin. You can take off this area called ZPA, which is where the digits will form. You can take that and you can place it onto another limb bud, and you can get multiple digits to form on opposite ends of that bud. Um, so that's where things like polydactyly might have, originated from, things like that. So pretty interesting, how susceptible we are to a lot of external factors and things that can influence the development process and how intricate the development process is in order for it to actually go correctly. So thanks for watching, and next time we'll be looking at how animals respond to their environment.